In today's lecture, I will be talking about inflammation. And as you know that the immune system has two arms, innate immune system and adapted or acquired immune system. The innate immune system is composed of four barriers: anatomic and physiological and phagocytic and inflammatory barrier. So inflammation is an immune response we're born with. But however, it is an augmented immune response. And as you know, that the aim of immune response is to remove the antigen or non-self antigens from the body. So, as you know that inflammation or the inflammatory process may follow several factors, for example, trauma or in some infection. And the inflamed area may show 5 cardinal signs: redness, hotness, tenderness, swelling, and the 5th one is loss of function. These signs, we call them cardinal signs. And these cardinal signs are due to 3 major events: increased capillary permeability. Here, these are the capillary lined with endothelial cells. So the permeability increased following inflammation, and it is also due to vasodilatation of the blood vessels, and as can be seen here, influx of cells. So influx of cells, increased capillary permeability, and vasodilatation, these are the 3 major events of the inflammation. And before that, I would like just to give an insight on the traffic of lymphocytes and maybe leukocytes. And as you know that lymphocytes, they have short stay in the circulation, around 30 minutes, and around 45% of these lymphocytes, they are carried to spleen and stay there for around 5 hours, and around 45 or 42% are carried to lymph nodes and stay there for 12 hours. And the aim for that is to encounter non-self antigens. However, 10% of the cells, and namely leukocytes, maybe neutrophils, they cross between endothelial cells line in the capillary, and there are some receptors in the wall of the blood vessel. These are receptors allow the white blood cells to settle first and to get out. So these endothelial cells, they express adhesions molecules that interact with the adhesion molecules on the surface of the white blood cells. And these adhesion molecules, they belong to four families. They are mucin-like. Here, for example, this is mucin-like on the surface, for example, of the neutrophil. And E-selectin is an adhesion molecule on the surface of the endothelial cell. And integrin, adhesion molecule, interact with immunoglobulin superfamily on the surface of the endothelial cells. These adhesion molecules actually allow these to be arrested, allow white blood cells to be arrested. And as you know that the blood, it goes in a central velocity. So these cells, in order to get out or to extravasate, they should follow 4 sequential steps. And these steps are rolling. So these include interaction between these adhesion molecules, then slow rolling, then the cells get arrested, then these cells extravasate and come out of the blood vessel of the capillary. Actually, this is helped by some cytokines, for example, tumor necrosis factor alpha, that induce the adhesion molecules on the surface of the endothelial cells. However, some of these adhesion molecules are constitutively expressed on the surface, but others, it needs some cytokines to express or to get expressed, and this is usually following an inflammatory process. And also some chemokines. These are cytokines, attract the neutrophils or white blood cells from the circulation to the site of inflammation. So during activation, the interleukin number 8 and macrophage inflammatory protein beta and TGF beta, transforming growth factor beta, these are chemoattractants for the white blood cells to the site of inflammation.